Hey, future pharmacists! Dr. Mike here. Nailing cellular and molecular pharmacology is like unlocking a secret level in your M.Pharm journey. It's the foundation for understanding how drugs work at the cellular level. Whether you're prepping for exams or diving deep into the world of drugs and cells, you're in the right place. In this video, we're tackling the most important questions in cellular and molecular pharmacology. We're talking about concepts that often trip students up, the ones professors love to put on exams. So grab your notes, get comfy, and let's conquer this together. Welcome to M.Pharm Masterclass. Let's begin. Alright, let's kick things off with the basics. Cell biology. Cells are the fundamental units of life, and understanding how they work is crucial for grasping pharmacology. Think of it like this. If you want to fix a car, you gotta know how the engine works, right? Same goes for drugs and cells. Organelles are like specialized compartments within a cell, each with its own unique job. You've got your nucleus, the control center that houses DNA, and the mitochondria, the powerhouse of the cell. The rough ER, studded with ribosomes, is all about protein synthesis, while the smooth ER is involved in lipid synthesis and detoxification. The Golgi apparatus packages and ships proteins, and lysosomes break down waste products. Cells are busy places. Now that we've covered the basic players in the cell, let's talk about gene expression. Think of your DNA as a giant blueprint, and gene expression is the process of reading that blueprint and building something from it. Gene expression is tightly regulated to ensure the right proteins are made at the right time and in the right amounts. We're talking about transcriptional regulation, post-transcriptional regulation, translational regulation, and post-translational regulation. Understanding this is key for understanding how drugs work, especially those that target specific genes or proteins. It's a complex process, but crucial for pharmacology. Let's move on. Okay, future pharmacists, let's talk about cell death. Cell death is a normal part of life and essential for maintaining a healthy balance in the body. There are different ways a cell can die, and understanding these differences is crucial for understanding disease and developing new treatments. First up, we've got apoptosis, also known as programmed cell death. It's a tightly regulated process essential for getting rid of damaged cells and shaping tissues during development. Then there's necrosis, which is more like an unplanned death, usually caused by injury or insult to the cell. Finally, we've got autophagy, a survival mechanism where cells recycle their own components during times of stress. Understanding these processes is key for pharmacology. Let's keep going. Chapter 4. Cell signaling. Deciphering the language of cells. Cells are like tiny chatterboxes, constantly sending and receiving messages. These messages are crucial for coordinating bodily functions, from growth and development to metabolism and immune response. Messages are often carried by molecules called ligands, which bind to specific proteins on the cell surface called receptors. When the right key fits into the right lock, it triggers a cascade of events inside the cell, leading to a specific response. There are different types of receptors, each with its own unique way of transmitting signals. Understanding these different receptor types is key for understanding how drugs work. Let's move on. Chapter 5. Secondary messengers. Amplifying the cellular message. Secondary messengers are small molecules that act as intermediaries, relaying the message from the receptor to other parts of the cell. One of the most important secondary messengers is cyclic AMP or CAMP. When activated, CAMP sets off a chain reaction, activating an enzyme called protein kinase A, leading to a cellular response. Another key player is inositol triphosphate, or IP3, which causes the release of calcium ions into the cytoplasm. Calcium is involved in all sorts of cellular processes, from muscle contraction to neurotransmitter release. Diacylglycerol, or DAG, activates protein kinase C, involved in regulating cell growth and differentiation. Understanding these messengers is crucial for pharmacology. Chapter 6. PCR, the DNA detective. PCR is like a DNA photocopier, allowing us to take a tiny amount of DNA and amplify it, making millions or even billions of copies. This is incredibly useful for diagnosing diseases, studying gene expression, and even solving crimes. Imagine you're a doctor and you suspect your patient has a bacterial infection. With PCR, you can take a sample, amplify the bacterial DNA if it's present, and voila! You've got your diagnosis. PCR is also a game changer for genetic testing, identifying mutations in genes that can cause diseases. In forensic science, PCR is used to analyze DNA evidence from crime scenes. It's a powerful tool transforming medicine and beyond. Chapter 7. Gene Therapy: Rewriting the Code of Life. Gene therapy is a revolutionary approach to treating diseases by correcting defective genes. There are different ways to deliver therapeutic genes into cells, such as using viral vectors or non-viral methods like liposomes. Gene therapy has shown great promise in treating a wide range of diseases, from inherited disorders like cystic fibrosis to acquired diseases like cancer. For example, in cystic fibrosis, gene therapy aims to replace the defective CFTR gene. In cancer, gene therapy can deliver genes that kill tumor cells or boost the immune system's ability to fight cancer. While still in early stages, gene therapy holds immense potential for treating challenging diseases. Let's continue. Chapter 8. Pharmacogenomics: Personalized Medicine at its Finest. Pharmacogenomics is the study of how our genes influence our response to drugs. Our genes play a big role in how our bodies break down and process drugs, affecting both effectiveness and potential side effects. Cytochrome P450 enzymes, or CYPs, are key players in drug metabolism. Variations in CYP genes can lead to differences in drug response. Understanding a patient's genetic makeup can help doctors personalize their medication choices. It's all about tailoring treatment to the individual patient. Let's move on. Chapter 9. Immunotherapeutics: Unleashing the Power of the Immune System. Immunotherapy is a groundbreaking approach to treating diseases by harnessing the power of the immune system. One exciting area of immunotherapy is checkpoint inhibitors, which block proteins that normally keep the immune system in check. By blocking these checkpoints, checkpoint inhibitors unleash the immune system to attack cancer cells more effectively. Monoclonal antibodies, or mAbs, target specific proteins on cancer cells, marking them for destruction by the immune system. CAR T-cell therapy is a personalized approach where a patient's own T-cells are genetically modified to recognize and attack cancer cells. Immunotherapy is revolutionizing cancer treatment, offering new hope for patients. It's a rapidly evolving field with immense potential. Chapter 10. Flow cytometry: Illuminating the Secrets of Cells. Flow cytometry is a powerful technique that allows us to analyze cells one by one as they flow through a laser beam. Cells are labeled with fluorescent markers, passed through a laser beam, and the scattered light and emitted fluorescence are detected and analyzed. Flow cytometry provides valuable information about cell populations. It's used in a wide range of applications, from diagnosing diseases like leukemia to monitoring the effectiveness of treatments. It's a powerful tool helping us understand the complexities of cells and develop new treatments. Let's wrap things up. Chapter 11. Biosimilars versus generics: Decoding the Differences. Generic drugs are identical copies of brand-name drugs that have lost their patent protection. They have the same active ingredients, dosage form and route of administration as the brand-name drug, but they're typically much cheaper. Biosimilars are similar to biologic drugs, which are large, complex molecules made from living organisms. Because biologics are so complex, it's impossible to make an exact copy. Biosimilars are highly similar to the reference biologic, but they're not identical. They undergo rigorous testing to demonstrate their similarity to the reference product before approval. Both biosimilars and generics are important for increasing access to affordable medications. Understanding the nuances of each is crucial. That's all for today's session. I hope these questions help you in your exam preparation. If you found this video useful, please like, share, and subscribe to M.Pharm Masterclass. See you in the next one. You've got this.